Hello, in the next couple of lectures we're going to talk about how the glomerular filtration rate in the kidneys is controlled, and there are two major sets of ways that this is controlled, and those include intrinsic control, so those are the things that the kidneys do automatically to make immediate changes to the glomerular filtration rate, and extrinsic controls, which means factors from the outside are going to control what's happening in the kidneys, and this can be important on a more of a longer term type basis. All right, just to review again, as you guys know, we've got blood that's coming into the kidneys from the renal arteries, and it gets distributed up into the cortex of both kidneys, and it winds up entering these afferent arterioles, and those lead to the glomerulus in each of these units that you have all over the place in the cortices of your kidneys. This is where glomerular filtration takes place, and then the filtered blood leaves through the efferent arteriole that we see over here. All right, so glomerular filtration rate is dependent upon how much blood is actually making it over into the glomerulus over here. All right, so more blood flow into the glomerulus is going to increase your glomerular filtration rate. Why? Because if you have more blood in here, then the hydrostatic pressure in there, the blood pressure, is going to be higher, and that's going to squeeze more plasma out of the glomerulus and over into the glomerular capsule. All right, so how do we control how much blood is actually making it from the afferent arteriole into the glomerulus? Well, a key way to do that is if you squeeze the afferent arteriole, if you constrict it, constriction of the afferent arteriole is going to reduce blood flow into the glomerulus. That makes sense, hopefully. If you pinch that afferent arteriole, less blood's going to make it over in here, and then that is going to reduce your glomerular filtration rate. Now on the flip side, if you dilate the afferent arteriole, more blood is going to make it into the glomerulus. That's going to increase the blood pressure in there, the hydrostatic pressure, and that's going to squeeze more fluid out from the glomerulus and into the capsule. We're going to have more of that glomerular filtrate that is produced. All right, so what we're going to see is that this is going to be your chief way for regulating production of glomerular filtrate and how much of that is made. And keep in mind, you got gazillions of these nephrons and glomeruli scattered throughout the cortices of both of your kidneys. All right, so as I mentioned, we're going to see it's very important for glomerular filtration to be controlled, because if you have too much of it, it could lead to loss of fluid volume in the body. If you have too little of it going on, you're not removing wastes properly from the body. Electrolyte levels can get screwed up. Fluid volumes can actually be too high in those cases. And so because of that, it's very important to control glomerular filtration very, very carefully. So it's tightly controlled, and the mechanisms that are used can be intrinsic, and these are things that happen instantly, instant adjustments, very fast adjustments that are made by the kidney itself. And then we also have extrinsic control. So this is where the nervous and the endocrine system get involved, and the mechanisms that the nervous system and the endocrine system influence really control blood pressure, but that blood pressure affects kidney function. In general, an increase in blood pressure leads to an increase in glomerular filtration rate. That's another thing to remember as well. That makes sense if you think about what's going on. If you have higher blood pressure, you're going to have more blood pushing its way into the afferent arterioles, and so you're going to have more blood flow into the glomeruli, so you're going to wind up with more glomerular filtration. If you have lower blood pressure, you're going to have less blood flow into the afferent arterioles, and then you're going to have less blood making its way into the glomeruli, so you're going to wind up with lower glomerular filtration. So that's another way you can control how much fluid is actually filtered out of the blood by controlling blood pressure. All right, so first let's talk about the intrinsic controls, which happen all the time within the kidneys to make sure that our glomerular filtration rate stays pretty much constant. So our bodies are really good under normal circumstances at keeping that glomerular filtration rate steady, no matter whether you're active, whether you're chilling out at home, or when you're sleeping. So the intrinsic controls of the kidneys will work across a very broad range of mean arterial pressure. That's what that means, like your average arterial pressure. When it's in the range of 80 to 100 millimeters of mercury, the intrinsic controls generally take care of things pretty well. And there are two major ways that this happens. One is called the myogenic mechanism, and that word myo refers to muscle. And so really what we're referring to here is the smooth muscle in the walls of arterioles. And then there's a second mechanism that we'll talk about, and that's called the tubuloglomerular feedback mechanism, which is kind of a mouthful to say, but it involves this juxtaglomerular apparatus. Remember, juxta, when you see that prefix, it next to. So this is that apparatus that's formed from part of the distal convoluted tubule, and it's very close to the glomerulus, and it also involves cells that are up against the walls of the afferent arteriole. So we'll see how all that works. Okay, so what's this myogenic mechanism? This is just a natural inherent mechanism of the smooth muscle cells that are in the walls of the afferent arterioles. All right, what happens? Let's say your blood pressure is going up. You have increasing blood pressure. That means more blood flow is going to cause the walls of the afferent arterioles to expand. Okay, and so you have smooth muscle cells in there, and when they sense that expansion, they naturally try to resist it because they're going to try to maintain a stable glomerular filtration rate. So if you think about it, if the walls of the afferent arterioles are expanding, that's going to increase blood flow into the glomeruli. So the smooth muscle cells that are located in the walls of the afferent arteriole will naturally contract and constrict the afferent arteriole. And what that's going to do is it's going to lower the blood flow into the glomerulus, and that will lower the glomerular filtration rate. So some of these things I'm just going to warn you with this glomerular filtration rate stuff. You're going to have to really take thorough notes and go back over and learn it as a story, because some of it can be a little bit complex. So be sure you're taking good notes and reviewing it pretty frequently. This is important because if you have blood pressure that's really going up high and you didn't have this myogenic mechanism, high blood pressure could really force a lot of blood into the glomeruli, and they're very delicate structures that already deal with a fairly high amount of pressure anyway under normal circumstances. And if they weren't protected from this increasing blood pressure, they could be damaged really, really easily. All right, so the opposite happens. Let's say you have falling blood pressure. That causes the smooth muscle cells in the walls of the afferent arteriole to relax. So what they're going to do is, you've got this falling blood pressure, so the walls are, getting kind of squeamish. There's not a lot of pressure up against them, and the smooth muscle cells in there will relax even more. That helps dilate the afferent arteriole. And dilation of the afferent arteriole increases the amount of blood flowing into the glomeruli. So that's the purpose of that. Okay, so that's your myogenic mechanism, and that just happens naturally because the smooth muscle cells in the walls of the afferent arteriole are able to sense these changes. Okay, and again, here's your afferent arteriole right in here, and again, you've got those smooth muscle cells in there that just automatically deal with those changes. All right, so the next type of intrinsic control involves tubuloglomerular feedback. And all right, so over here on this part of this diagram of we've got our renal corpuscle there, which is the glomerulus and the glomerular capsule, and proximal convoluted tubule. Here's our loop of Henle, or our nephron loop, and then here's our distal convoluted tubule over here. Okay, and so this juxtaglomerular apparatus is located right around in there. That's what's blown up over here. And what you're seeing right here, that is a cross section through, now the diagram is telling us it's part of the ascending limb of the loop of Henle, but you can see it's also that part of the tube there is already forming some of the distal convoluted tubule sections, but that's what you're seeing cut open there. And these cells that are highlighted in purple here, those are called macula densa cells, and they play a very important role in the function of this juxtaglomerular apparatus, and also helping the kidneys maintain proper glomerular filtration rate. And then the other cells we'll really be talking the most about over here are these granular cells. And so they're located right in here, and you've also got some over here. The granular cells secrete renin. And so we'll be hearing about them as well as we talk about these topics. And then you've got these extraglomerular mesangial cells. And they may play roles in communicating the signals between the macula densa cells and the granular cells that you see there on the diagram. All right, so a little bit more about that juxtaglomerular apparatus. One per nephron, and it's very important, importantly involved in regulating the formation of our filtrate, controlling what's present in this urine that's developing, and blood pressure as well. And again, as we were taking a look at, it involves, you got those macula densa cells in there that are part of the distal portion. Again, proximal means you're closer to the renal corpuscle. Distal means you're farther away along that pathway from there, even though they do come back together and form this structure. So the distal portion of the ascending limb, where it's kind of becoming the distal convoluted tubule, and also part of the and also these granular cells that are located next to the afferent arteriole. There's another name for those. They can also be called JG cells or juxtaglomerular cells. Those cells contain granules. The name granular, if you look at them under the microscope, they would look spotty on the inside. And the granules are little storage compartments for renin, the good old renin that we heard about when you guys were learning about blood pressure, the renin-angiotensin story. Another thing these granular cells can do is they can function as mechanoreceptors. That means they're stretch receptors. And so if blood pressure is going up, these granular cells can tell the afferent arteriole is getting stretched like this, and so they're able to sense that and make adjustments accordingly and take action according to what they're sensing. The macula densa cells we were taking a look at on the diagram, they are tall cells like they look as they appear on the diagram from the textbook, closely packed together. they act as chemoreceptors. And what they're going to do is they're going to sense the sodium chloride content of the filtrate. All right, so remember where they're located, this urine that's forming has passed through the proximal convoluted tubule where you've already had a lot of reabsorption has taken place. It's gone down through the loop of Henle, it's coming back up, and it's all the way up at the top at the very end of that ascending limb. And by now you're getting pretty close to having taken out all the stuff you need to take out of the urine and put in extra things that you need to put in through tubular secretion. So you're getting pretty close to having fully formed urine. And so these macula densa cells, what they're looking at is, what's the sodium chloride content? So we've heard of chemoreceptors before that are sensing things like carbon dioxide concentration in the blood, oxygen concentration in the blood. These cells can detect changes in sodium chloride content. So they're looking to see whether it's too high. Okay, if it's too high, that's an indicator is too high. Because what's going on, if your GFR is too high, then you're pushing this fluid through these nephrons too quickly. And so there's not enough time for the sodium ions and the chloride ions to be reabsorbed properly. On the flip side, if the sodium chloride content is too low, that's an indication that your GFR is too low, and too much sodium and too many chloride ions are being reabsorbed back out of the filtrate as it passes through the nephrons. So checking out that sodium chloride content in that part of the nephron is a pretty good method for determining whether or not, for the kidneys, an intrinsic mechanism for them to determine whether the GFR is too fast or too slow. All right, so I think this slide is just summarizing for you in a text format what I just mentioned. So these macula densa cells sense sodium chloride concentration in that filtrate that's passing by them. All right, and if your GFR is too high or it's too fast, then your sodium chloride concentration will be high because there's insufficient time for reabsorption of sodium ions and your chloride ions. Okay, sorry about that. I had a little communication problem again between iPad and computer. For reabsorption of sodium ions and chloride ions. On the flip side, if the sodium chloride concentration is too low, that means the glomerular filtration rate is too low and things are proceeding through the nephron too slowly. So too much sodium and chloride is being reabsorbed back out of the filtrate, and the concentrations of those two ions will be low. All right, so if the macula densa cells I'll back up here. If the macula densa cells detect increased sodium chloride, and again that means our GFR is too high, they secrete various vasoconstricting chemicals, and those act on the afferent arteriole. Okay, so if the afferent arteriole constricts, less blood is going to flow from the afferent arteriole into the glomerulus. That's going to reduce your GFR. So this will probably be happening along with the myogenic mechanism, where the afferent arteriole is by itself constricting. It's detecting the stretching, and it is by itself constricting anyway to help limit the flow of blood from the afferent arteriole into the glomerulus. That decreased GFR will slow down the flow of the filtrate, allows more time for reabsorption. And again, as I mentioned, the opposite occurs if GFR decreases. If that's the case, then these MD cells over here won't release these vasoconstricting chemicals. Also, that the myogenic mechanism will be involved as well. And instead of constriction, you'll have dilation of the afferent arteriole, which increases the flow of blood into the glomerulus and increases the GFR. So since we've got those two intrinsic mechanisms in the kidneys working so well across both kidneys if you're healthy, it's probably not a surprise then that your glomerular filtration rate can stay pretty constant, no matter whether your mean arterial pressure is 80 or whether it's 180. If you're anywhere within those ranges, then things should be functioning normal. Now, if you go too high, the intrinsic mechanism can't handle things, and if you go too low, the intrinsic mechanisms by itself can't maintain proper glomerular filtration rate. All right, so in the next video lecture, we're going to talk about the extrinsic controls. This is where your nervous system and the endocrine system get involved in helping to control glomerular filtration rate.